Hello, everybody. This is our presentation for Project 1. I'm Carly.
I'm Kirsten.
I'm Kelsey.
And I'm Hannah.
So the purpose of this experiment was to determine how peritonitis affects different time points during the immune response. We hypothesized that for 0, 2, 5, and 24 hours, the amount of adaptive immune cells will increase respectively depending on the time period of peritonitis. We also expect to see the same pattern in the blood differentials. Peritonitis is the inflammation of the peritoneal cavity, and thioglycolate is a medium used for cell structure. Here it is used to induce peritonitis, causing inflammation, which in turn causes a surge of neutrophils and monocytes. Other immune cells that play a role in the immune response are monocytes, neutrophils, lymphocytes, T and B cells, eosinophils, and basophils. So some of the materials we used were mice, thioglycolate, the flow cytometer, and blood smears. We had treatment levels at the 2, 5, and 24-hour time points and a control at 0 hours. And for each of these levels, we had 3 mice. The mice treated with thioglycolate were injected intraperitoneally, and the data from the controls and treatment levels were collected from peritoneal fluid and blood smears. The blood differentials and flow cytometer measurements were used to determine the amount of immune cells that were present.
So for our results, figure 1 shows that the average percentage of white blood cells at various time points of the peritoneal injection. Cells from blood smears were looked at under a microscope to determine the type of each cell. Percentages were then calculated at each mouse and averaged at the different time points of exposure. Monocyte counts were significant with a 0.005 p-value at 0 versus 2 and 0 versus 24-hour time points. Monocytes were also prevalent at the most prevalent at the 0-hour time point. Lymphocytes were significant with a 0.0 p-value at 2 versus 5-hour and 2 versus 24-hour time points. Lymphocytes were the highest among all of the time exposures. Segmented neutrophils were significant with a 0.0 p-value at the 0 versus 24-hour and the 2 versus 24-hour time points. Banded neutrophils were significant with a 0.005 p-value at the 0 versus 2-hour and the 2 versus 5-hour time points. These were also significant at the 2 versus 24-hour time point with the 0.05 p-value. Banded neutrophils peaked at the 2-hour time point. Figure 2 shows the average percentage of each cell type obtained from the peritoneal cavity. The percentages were calculated from flow cytometry data from each mouse and averaged for each time point. Macrophages were significant at the 0 versus 2-hour and the 0 versus 5-hour time points with a 0.005 p-value. They were also significant at the 0 versus 24-hour time point with a p-value of 0.005. At the 2 versus 5-hour time point, they were significant with a 0.05 p-value. Neutrophils were significant with a 0.05 p-value at the 0 versus 2, 0 versus 5, and 0 versus 24-hour time points. Neutrophils peaked at 2 hours, then began to decrease. B cells were significant at the 2-hour versus 5 time point with a 0.05 p-value. T cell results were not significant. Macrophages and B cells dropped from hour 0 to hour 2, then began to increase from hour 2 to hour 24. Figure 3 shows the average total number of cells obtained from the peritoneal cavity. The number of cells were calculated through flow cytometry and averaged for each mouse at each time period. Mouse 2 from hour 2 and mouse 1 from hour 24 were excluded from this figure. The only significant value was a p-value of 0.005 between hour 0 mice and hour 5 mice. The number of cells was lowest at hour 0. After hour 0, it began to increase with a sharp increase in cells for mouse 3. The cell count initially increased with time, except for mouse 3 that decreased with time. Figure 4 shows the average total number of each cell in the peritoneal cavity. Each cell type was found by flow cytometry and calculated for each mouse at different time points. Macrophages were found to be significant with a 0.05 p-value at the 0 versus 24-hour time point. Neutrophils were significant at the 0 versus 5 and 0 versus 24-hour time points with a 0.005 p-value. There was a sharp increase in neutrophils between 0 and 2 hours, then they decreased from 2 to 24 hours. B cells were significant at the 0 versus 5 and 0 versus 24-hour time points with a 0.005 p-value. T cells were significant with a 0.05 p-value at the 0 versus 5-hour time point, and the average number of T cells increased between hours 0 and 2, then decreased from 2 to 24 hours. The amount of macrophages in B cells slowly increased over time.
From our flow cytometry data, we see that our hypothesis was half right. We saw that the B cells followed our hypothesis and increased after the injection. The T cells, however, didn't follow our hypothesis and decreased after some time. From our blood differentials, our hypothesis was correct despite the inconsistent trend, and the lymphocytes increased from hour 0. The blood differentials and flow cytometry data show different results, but this can be attributed to the subjectiveness of blood differentials and the objectiveness from the flow cytometer. There can also be inconsistencies due to the unrecorded amounts of fluid retrieved from the peritoneum. On this slide, we've compared our study to 2 additional studies. The left one shows a similar finding, but over the span of days, not hours. We see an initial spike in neutrophils and then it serpentines before leveling out over the following days. We also see a similar trend in T cells where they are first low and then we would expect to see an increase over time. On the right, we see a study that disagrees with our study, and it claims that we should have seen an upward trend of neutrophils, but instead we saw an initial spike and then a fall. And this could be due to the use of different mouse strains as well as a different choice in the peritonitis induction.
So this is showing the pathway of the newly developed neutrophils. They start in the bone marrow and mature, and then they're transferred into the circulatory system, where they are able to recognize thioglycolate, and they travel to the peritoneal cavity. Then they phagocytose the thioglycolate there and call for help from more cells. This is relevant because thioglycolate has been shown to cause a surge of neutrophils, which help recruit lymphocytes. So in our study, we used a mouse model to mimic the inflammatory response in the peritoneum, and looking at different stages of the infection, different immune cells can be expected. So this would affect what treatment was needed.